Good morning everyone. I am Emlata Kanniappan doing my research under the guidance of Dr. Vignesh Muthuvijayan. Today I will be giving a lecture on immunology. The main lecture is based on immune responses to foreign materials or biomaterial or any implant. So before we understand or go deep into the understanding of immune responses in the presence of biomaterial, we should know few basics of immunological responses or immunological terms and or what is immunology or what are the cells involved in immune responses and all. So in this lecture I will be giving a brief introduction about what is immunology or what are the immune responses or what are the types of immune responses or what are the cells or organs involved in immune system and the major cells which are responsible for immune responses. So we should know what is immunology. So what is immunology? It is just the study of biology which studies the immune systems of all organisms. Or what do you mean by immunity? We say, right, you have a very good immune response for this kind of infection or so what do you mean by that? So what is immunity? Immunity is nothing but the protection of body against pathogens or infections caused by pathogens. When I say pathogens are like viruses, bacteria, fungi, parasites or some proteins. So this is about immunity and what is pathogen, and there are two types of immunity, innate immunity and adaptive immunity. When I say it is innate immunity, it is the primary form of defense mechanism in our body. So our body it itself has a defense mechanism, like it is a very good it has a very good defense force to act against the pathogens which enters the body. So it is divided into innate and adaptive immunity. So innate immunity, where innate immunity is like it is always on. Okay, it is present from the birth. This is the primary form of defense mechanism. So always on, which means it has barriers. When I say barriers, it means what? What are the barriers that protect our body from the pathogens? The first and foremost is skin. Skin is the largest organ of the body. It protects our entire body from various pathogens entering into our body. The next one is mucous membrane. Mucus. There are several places in our several parts of body are covered by mucus which protects those parts from the pathogens. Mucus are nothing but the two or more layers of epithelial cells. And then pH, the acidic pH of our stomach, which is like around 2 to 3, very acidic, which kills the bacteria effectively. Skin, mucous membrane, pH, then microbial flora, which is present in our body. There are some good bacteria in our gut which protects our body from the pathogens. So these are the main barriers present from our birth for to fight against the pathogens. Then again in innate immunity, this part of the cells are always present, okay, always on. No matter what the pathogen enters, it will go and kill that, it will go and act against that pathogen. The next is immediate response, where the cells present in this like they are not activated all the time, but once when the pathogen enters, it immediately gets activated and start acting or fight against the entering pathogen. For example, blood cells. So these blood cells, it will be there in the it will be there in the bloodstream. Once when the pathogen enters, it will get activated and fight against those pathogens. And one more thing, in always on, kind of immunity, there is one complement factors. Complement factors are not the cells, they are kind of proteins which acts against the pathogen, not cells, okay, which acts against the pathogen. So, this is about the innate immunity. And the next one is acquired immunity or adaptive immunity. It is more specific and specialized kind of response which becomes part of our immune system at a later stage, which means it is not an immediate response, it is a kind of delayed response. Okay. So, adaptive immunity is delayed response. So, the major cells responsible for this type of immunity is lymphocytes. Lymphocytes again divided into T cells and B cells and natural killer cells. T cells, B cells and natural killer cells. Okay. And this B cells produces antibodies. These B cells produces antibodies which fight against the pathogen, and T cells again T helper cells and T cytotoxic cells are there. Okay. Natural killer cell distinguish from T cells and B cells with the presence of where B cells and T cells are the specialized receptors on the surface, natural killer cells will not have that. That is the major difference between these cells. Okay. So, when I say the immunity is based on the cells, because of the cells, where in innate immunity or as well as in the adaptive immunity, when I say the immune response is because of the cell, it is called cell mediated immune response. Cell mediated immune response. The other immune response is mainly based on proteins or these antibodies, they are not cells, which are termed as humoral mediated immune response. Okay, these are the two major immune responses which acts in our body. So, in this slide where innate immunity is non-specific resistance, which means no matter what the pathogen is, once it enters the bloodstream, it will go and engulf and it will kill the pathogen. That is non-specific immunity, whereas the adaptive, it is more specific, it will go and kill the most specific antigen or a specific, it will produce a specific antigen to kill the specific pathogen. Okay, specific resistant responses to the immune system. So, the first line of defense, we all know that the barriers. So, the barriers are nothing but skin, mucous membrane, where for example, for mucous membrane is like nose, eyes, lips and all, where eyes is covered by mucous membrane, it is not covered by skin, which protects our eyes from the external pathogens. And normal microbiota present in our gut. Our gut will produce a good bacteria to engulf the bad bacteria. Also the pH. And the second line of defense are the phagocytes which engulf the microorganisms mainly of WBC cells. And the third line of defense, which are the adaptive immunity, specialized lymphocytes. The lymphocytes are the major cells responsible for this central cells for the adaptive immunity, which are again divided into B cells and T cells, and the B cells produces antibodies which fight against the pathogen. So, these are the specific, non-specific OS defenses. Summary of non-specific innate immunity. Anatomic barriers, which are skin, mucous membrane. The skin is the largest organ of the body and it plays a very important role in our defense mechanism, and mechanical barrier which stops the entry of microbes on the first step itself, and the acidic environment, stomach acidic, which digests the bacteria, and mucous membrane again helps to protect the entry of microbes into the internal organs, and the physiological barriers like temperature, low pH, that which I said already, acidity of stomach, and chemical mediators or the factors. complement factors, and phagocytic or endocytic barriers, where the various type of cell which will endocytose is nothing but the engulfing the cells and breaking down into foreign macromolecules, and the cells responsible for those are macrophages, neutrophils, monocytes, neutrophils and tissue macrophages. An inflammatory barrier, where the tissue damage and infection induce leakage of vascular fluid. This contains the serum proteins which has the antibacterial activity and into the affected area. So, this is the principal mechanism of innate and adaptive immunity. In terms of adaptive immunity, the adaptive immune system has 4 important characteristic attributes. The first one is antigen specificity. These are the characteristics of adaptive immunity. The second one is diversity. Third one is immunologic memory. And the last one is self, non-self recognition. Non-self recognition. First attribute, characteristic attribute is antigen specificity. So, it permits the immune system to distinguish even a small difference among the antigens. So, it will be more specific in recognizing the antigen. And diversity, it allows the immune adaptive immune system for generating tremendous in diversity of recognition molecules in allowing that to recognize billions of unique structures present in the foreign antigens, that is diversity. And immunologic memory, once the immune system recognized the antigen and responded to the antigen, when the same antigen it encounters the second time, for the second time it will develop the high level of immune response, to that specific pathogen. It keeps that in its memory that we have encountered this already and we became immune to that. So, this is the this attribute is a very important attribute in adaptive immunity. And finally, once it is responded, and the immune system should be able to differentiate from the self and the non-self components in order to act on the non-self components. So, what will happen when the immune system fails to recognize between or differentiate between the self and non-self components? Yes, obviously it will lead to some other responses and it could be fatal. So, these are the major 4 important characteristics of adaptive immunity. So, the principal mechanisms of adaptive immunity, so main is microbes, epithelial barriers, phagocytes and complements. And in case of adaptive immunity, B lymphocytes and T lymphocytes. B lymphocyte produces antibodies and T lymphocytes are the effector T cells, Th cells and Tc cells. So, this is all about the types of immunity. Like to summarize that, innate immunity and adaptive immunity. Innate immunity is non-specific resistance, like whatever the pathogen it enters the body, it will go and act on the fight against that pathogen. Whereas the adaptive immunity, it's not an specific, it's like completely specific to the pathogen and fight against that pathogen, and it is also termed as delayed response. Adaptive immunity is also a delayed response, termed as delayed response immunity. Whereas innate immunity, it acts as immediately, there is immediate response as well as always on response. There will be some barriers which are always on in the innate immunity, and major cells present in innate immunity is like skin, the epithelial cells and microbes, and there are and few chemical factors or physical factors are also involved, like temperature or pH and chemical cytokines, and those are involved in innate immunity. Adaptive immunity, major cells in adaptive immunity are lymphocytes. Okay. So, this all about immunity. Now, we will go to the cells present in the immune system, cells of the immune system. So, and this picture is the process of hematopoiesis. Hematopoiesis is nothing but the formation of blood cells. So, any blood cell will originate from the hematopoietic stem cell. Blood stem cell is nothing but hematopoietic stem cell, which is self renewing cells. Okay. And this self renewing cells will give rise to either of myeloid progenitor cells or lymphoid progenitor cells. These are progenitor cells. When I say they are progenitor cells, it will which means it loses the self renewing capacity, and it will be specific to a particular cell lineage. Okay. And myeloid stem cell progenitors, progenitor cells, lymphoid progenitor cells, and this hematopoietic blood cell originated from bone marrow. So, that is the place where this hematopoiesis takes place. Two major cell lineages is lymphoid cell lineage and the myeloid stem lineage. So, myeloid stem cell progenitor cell will give rise to granulocytes, progenitor cells, basophil progenitor cells, eosinophil progenitor cells. From the granulocytes, it will give rise to neutrophils, monocytes, and this monocytes give rise to macrophages, okay, which are very important cells responsible for immune system, immunity. And neutrophils, monocytes, macrophages, and neutrophil progenitor cells give rise to neutrophils, and basophil progenitor cell give rise to basophils, eosinophil progenitor cells give rise to eosinophils. Okay. Also erythrocyte progenitor cells, which give rise to red blood cells, and megakaryocytes, which gives rise to platelets. Okay. So, this is this all about the myeloid lineage. It also gives rise to dendritic cells. Okay, dendritic cells are antigen presenting cells. They are APCs, antigen presenting cells. Okay. And this all about myeloid stem cell lineage, which is nothing but which gives rise to granulocyte progenitor cells, basophil, eosinophil, and granulocytes again give rise to neutrophils as well as monocytes. From monocyte macrophages are originated, and it also gives rise to megakaryocytes, from which platelets are platelets originates, and then erythrocyte, from which RBCs. This all about myeloid lineage. Whereas in lymphoid stem cell lineage, where it gives rise to lymphocyte, which are again divided into B lymphocytes, T lymphocyte, and natural killer cells. Okay. Also it gives rise to dendritic cells. So, these are the again these are, we all know, these are the WBC cells, white blood cells. So, this natural killer cells mainly differ from B lymphocytes on the T lymphocytes with the presence of surface, the presence or absence of surface receptors, where in B cells and T cells have the presence, have the surface receptors, whereas in natural killer cells it do not have, and it is called as the large cell. It is a large cell, and whereas B and T are small naive cells. Okay, these are about the cells of the immune system, and we should know about the before going in depth with the cells, I will tell about the organs of the immune system. Okay. There are it is divided into primary organ, primary and secondary. Okay, in primary, bone marrow, and thymus. These are the organs responsible for the production of these cells, and secondary lymph node and spleen. Okay. So, these primary organs where it is the site for the maturation of the cells, B cells or the T cells. Okay. This is the maturation site. Once it got matured, it acts against the pathogen, right? It will go and fight against the pathogen that will act in the secondary organs, which are action site. This is a basic few points you should know when you study about immune system. The organs of the immune system, which are the primary lymphoid organs or the secondary organs. In primary, which is the place for the maturation of the cells, bone marrow and thymus for B cells and T cells, and the secondary organs are lymph node and spleen, where it will be an action site for the matured cells to act against the pathogen. So, this all about the organs and the cells of the immune system, and the morphology and staining characteristics of the various type of blood cells. So, from this the large lymphocyte, as I said in the previous slide, the large lymphocytes are nothing but natural killer cells, okay, and the small lymphocytes are nothing but B cell and T cell. Okay. And these are the small cells or the red cells or the red blood cells, and monocytes are also present, and basophils, eosinophils are also present. So, both red blood cells and platelet, they lack nucleus, and they are the most numerous cells present. Okay. Most numerous of the leukocyte populations are neutrophils. In the next slide you will be seeing the percentage of different cell types present in the WBC, and lymphocytes are the predominant cell type responsible for the immune response. Okay, they are the central cells responsible for the immune response. So, this slide will tell you about the percentage of cells present in the white blood cells. So, neutrophils it is like it occupies like 60 to 70%, okay, and basophils and eosinophils, basophils 0.5 to 1%, eosinophils 2 to 4%, monocytes 3 to 8% and lymphocytes 20 to 25%. And in this, neutrophils and eosinophils are again phagocytic cells. Neutrophils and eosinophils. Basophils are non-phagocytic. Okay. Monocytes give rise to macrophages. B cells, T cells. Okay. So, in this classification is mainly based on the granulation and the staining characteristic of cells. So, A granular and granular, it is based on the presence of granules in the cytoplasm, whereas also based on the classification of the nuclei, okay, based on the classification of morphology, based on the difference in morphology and staining characteristic. Or granulation. So, lymphocytes and monocytes are A granular, it does not have granules in the cytoplasm, whereas basophil, neutrophil and eosinophil has granules in the cytoplasm, and neutrophils are as a multi-lobed nuclei, okay, and it will stain with both acidic and basic dyes, stains both acidic and basic dyes. Okay. And eosinophils is it has a bilobed nucleus, where it stains only with the acidic dye eosin red, that is why its name as eosinophils, only acidic dye, it stains with only acidic dye eosin red, and whereas basophils stains only with basic dye methylene blue. Okay. So, this all about the classification based on the morphology and staining characteristics or granulation present in the cells. And this picture summarizes all the cells present in the immune system which are responsible for the immune responses. So, cells of the immune system, for innate immune system, for adaptive immune system, for certain cells which are produced in both innate and adaptive immune system, whereas neutrophils, monocyte, eosinophil, basophils are present in innate immune system, whereas T cell, B cell. So, when I say it is an innate immune system, these cells will be there always on, and when pathogen comes, it will go and act against the pathogen no matter what the pathogen is or what the antigen is. But whereas when I say the cells of adaptive immune system, they are the delayed response, they act against the specific antigen, which are T cell, B cell and T helper cell and T cytotoxic cells, and both immune system has macrophages and dendritic cells. Macrophages for the phagocytic activity. So, with this slide we know about what are the immunity and what are the immune responses and what are the cells present in the immune system and what are the organs of the immune system. So, next I will talk about macrophages. So, macrophages are nothing but we can say the enlarged monocytes. Okay. Monocyte circulate in a bloodstream for a period of 8 hours, during which they become enlarged and go to a specific tissue where they were fused together and enlarge and become a macrophage. Okay. Macrophage. So, the main difference between monocyte and macrophage is in the structure. Okay. The structure of macrophage is 5 to 10 fold larger than the monocytes. Larger than the monocytes. Also, the number of organelles and its complexity is increased than the monocyte, especially lysosomes. The number is increased a lot as well as its complexity. Also, it produces hydrolytic enzymes. So, this is the major difference between macrophage and the neutrophils. Neutrophils will not produce the hydrolytic enzyme, other than that it will go and phagocytize in the similar way how the macrophage do. But the macrophage secretes hydrolytic enzymes and some soluble factors as well as. So, macrophage is the fused enlarged form of monocyte. Okay. When it migrates and deposited to a specific tissue, it becomes a tissue specific macrophage, and these are the same images of the macrophage. The first is the macrophage same image where it has an like very long pseudopodia and it acts against the bacteria, right, and this is the macrophage, pseudopodia, bacteria. The same is here, but this one is our erythrocyte. So, how beautiful it is, the macrophages are, right. Macrophages are very beautiful, no, because they are very important to our immune system to fight against the pathogen. We should praise them, of course. And specialized macrophages which go and deposited in the tissue and it becomes the tissue specific macrophages, and here we have given the few examples of tissue specific macrophages. So, the macrophage in the lung are alveolar macrophages, and Kupffer cells in the liver, osteoblasts in the bone, microglia in the central nervous system which is in the brain, histiocytes in connective tissue, mesanglial cells in the kidney. These are the tissue specific macrophages. And this diagram shows the maturation of macrophage. First the stem cell and the monoblast, to that it forms an monocyte. Monocyte originate from this monoblast, and that monocyte fused and forms an enlarged one macrophage. Once it activates, it is an activated, there are two types like resting macrophages and activated macrophages. One resting macrophages, it will go and sit in one place, it will take rest. Once someone calls it, like when it gets some signal or then it will act against the pathogen. But certain macrophages are active throughout. Activated macrophages are more effective than the resting macrophages. That obviously, right, when like a person who is very active is more effective than a person who sits in the same place for all the time. And these are the tissue specific macrophages, and the lower picture, lower panel shows the activated macrophages. You can see how large it is. The activated macrophage in terms of morphology, also the presence of organelles in number as well as its complexity, the complex structure. So, activated macrophages are very effective than the resting macrophages. So, activated macrophages are more effective than the resting macrophages in killing pathogens for several reasons. They are like, it has increased phagocytic activity, and it produces increased production of inflammatory mediators, also it produces special cytotoxic proteins. These proteins can act against several pathogens, like specific pathogens like cytotoxic proteins. It acts against certain broad range of bacteria. It acts, this cytotoxic protein like acts against the infected, this cytotoxic protein acts against the broad range of targets including intracellular bacteria, virus infected cells, including intracellular or cancer infected cells, intracellular bacteria, virus infected cells or cancer cells. So, these are all about the activated macrophages. So, normally in resting state, macrophages are activated by a variety of stimuli in course of immune response. So, there are certain macrophages which will be in a resting state, which starts to activate once it gets the signal from the other cells, and phagocytosis, it is an important activating stimulus. Okay, phagocytosis is the important activity. Phagocytosis is nothing but the engulfing of the pathogen. So, again the macrophages are further activated by cytokines secreted by T helper cells. So, in this way, in the entire immune responses or immune system, this macrophages and T helper cells facilitate with each other, macrophages and Th cells, and they are the mediators by the, also it activates stimuli, mediators by the response, inflammatory response. These are the certain stimulus for the resting macrophages to get activated. Next is the phagocytosis. Phagocytosis are nothing but the engulfing of the foreign material. Okay. There are three steps. First, it has to go and recognize what is the pathogen, and then the neutrophil gets attached and engulf. Thereby, once it engulfs, the lysozyme and everything, all the enzyme acts on it, kill it and degrade it. So, engulfment and degradation may not happen with the biomaterials. Yes, right, the biomaterials will be large enough for that the phagocytes go and engulf it. So, it cannot do that for the biomaterials. It is a different type of phagocytic, we cannot say a phagocytic, it is a foreign body reaction will happen when in the presence of biomaterials. Foreign body reaction will happen in the presence of implants or biomaterials. So, these are the steps of phagocytosis for the engulfing and pathogen. The first, bacterium becomes attached to a membrane called pseudopodia. Then once it is attached, it is ingested from a phagosome, and then phagosome fuses with lysosome, and this lysosomal enzyme digests the captured material. Finally, the digestive products released from the cells. But if this target, if a phagocytosis fails to engulf the target, it will become more frustration. See, if I have given some work to do, if I didn't do that, obviously my senior or the guide who gave me this work will become frustrated, or even me as well, I will become frustrated, right. So, the same thing will happen to phagocytosis, because it cannot do the work what it is given to him. So, that is why it is termed as frustrated phagocytosis. Okay. So, now I will be showing a small video about the process called phagocytosis. What are phagocytosis? The engulfing of the bacteria by the immune cells. So, what are the immune cells which play a very important role in phagocytosis? Macrophages. So, now look into the video, you will understand even more better. So, those are the cells or macrophages which engulfing the bacteria. They eat and destroy the bacteria. They are the white blood cells which are called as macrophages. See how they are invading, it is very beautiful, right. So, the main job is to eliminate foreign entities that invade your body, which is through the process called phagocytosis. See, this bacteria, as soon as it enters, it releases some proteins which go and alert the macrophages to start its function. These macrophages attaches to that bacterium with the help of special proteins that are present on the cell surface. Then the bacterium is engulfed and locked inside a structure called phagosome. Then it is cleaved and destroyed by the digestive enzymes into pieces. And these resulting particles can be either used by the cell or get out of the cell, released out of the cell. And there it leads to the foreign body giant cells. The foreign body giant cells are the large cells which are formed as a result of fusion of macrophages. Okay, that happens in the presence of biomaterials, as I explained before. So, biomaterial cannot be engulfed by the phagocytosis. So, if any implant or any biomaterial are placed inside, there are series of reactions will happen. Okay, in that reaction, foreign body giant cell plays a very important role in that. So, foreign body reaction, if I say foreign body reaction, it is mainly due to the presence of macrophages and the foreign body giant cells. Okay. So, again the blood tissue and tissue biomaterial interaction and the biomaterial. So, see, monocytes, tissue is there, blood and tissues, and when I place a biomaterial, there will be some interaction between tissue and biomaterial, right. So, their macrophages will there, and in the biomaterial foreign body giant cells will form. So, the huge mass surrounding the biomaterial with the presence of macrophages and foreign body giant cells. So, foreign body giant cell is the collection of fused macrophages in the presence of, because of the presence of foreign body, okay, large foreign body. So, the adhesion of macrophages and foreign body giant cells at the surfaces of biomaterial exists between produces an environment that exists between the cell membrane and surface of the biomaterial. So, I will ask you one question. So, if I want to place a biomaterial, what are the factors you need to consider for an immune response? For an for an advantageous immune response, there are, see, we cannot directly place a biomaterial inside our body without a proper understanding of the immune responses or the proper understanding of the properties of the biomaterial, right. So, what are the properties you need to think a lot for an in vivo application or as an implant? Main thing is size and surface chemistry. Surface chemistry of a biomaterial, any material, biomaterial, if placing inside our body or placing in vivo is very important. Okay. Surface chemistry, so there should be some compatibility, right, compatibility between tissue and the biomaterial. See, there are two interaction majorly takes place, cell-cell interaction, cell-tissue interaction, cell-cell interaction and the tissue biomaterial interaction. tissue biomaterial interaction should be proper, that then only it become non-toxic. Otherwise, compatibility will be of question mark. Compatibility means the safety. Okay. So, you need to consider the surface chemistry, degradation of degraded products, like whether the degrading products or the leachate from the biomaterial should be non-toxic, and the size, surface chemistry, geometry, everything you need to be considered for the for degrading the immune response. So, inflammatory response. So, inflammatory response, it is the localized non-specific response to an infection or wound. It is just the physiological response as a result of infection or injury. That is the inflammatory response, the immediate response in our body. Tissue damage which are due to the pathogen invading pathogen or a wound, which induces the complex sequence of events that stimulate the immune response, right. That is collectively termed as inflammatory response. If my tissue is getting damaged, there will be a sequence of events takes place in order to repair this tissue. So, because our immune system is more effective by its own, right, to repair the tissue. So, that collectively known as inflammatory response. The inflammatory response is this complex sequence of events that happens when the tissue is injured or damaged. Okay. And this provides early stage of protection, early stage of protection by restricting the damage to the site of infection or tissue injury. So, tissue damage and infection induces the leakage of vascular fluid that containing serum proteins with antibacterial activity and inflects the phagocytic cells into the affected area. So, when there is any injury or any infection, so the first and foremost response, the inflammatory response will be the swellness, right, the redness or the swelling or heat or pain. We would have felt it at many times. By this time you guys would have felt it in many stages or at many events about the inflammation. See, if even for a small example, when a mosquito bites you, immediately it becomes swell, right, it swells immediately and you will get kind of pain. Again, it is an inflammatory response. Okay. And these are the redness, swelling, heat and pain are the cardinal signs of the inflammation are due to the three major events. These are the signs of the inflammation of the inflammatory response. Okay. So, when I say the events like injured cell which release chemical, once the cells got injured, immediately it starts secreting some cells, okay, signals. It is the kind of signal for the others to get activated, so that say think that yeah, it is got damaged, we have our duty to go and repair that. So, it releases the chemical alarm signals like they are called as inflammatory mediators, okay, histamines, prostaglandins and kinins, inflammatory mediators. So, these are the what to say medical indication to say that there is some injury or infection. Okay, histamine, prostaglandins or kinins that cause the blood vessels to dilate, which is the vasodilation. As a result, there is an increased blood flow to the site of injury. So, once the site got injured, there will be increased blood flow, that is vasodilation. Again, redness and swelling of the affected area due to increased blood flow. Also, there is an increase in tissue temperature. We would have felt no, there will be some heat generating if there is some injury, right, that is because of the increased temperature, tissue temperature at the affected site. And edema is the accumulation of fluid. Increased blood flow stretches blood capillary walls, increasing their permeability. This facilitates the influx of fluids and cells from the capillary to the tissue. So, this is the accumulation of fluid causes tissue swelling. Okay. So, edema is nothing but the accumulation of fluid and the influx of phagocytes. Increased permeability, influx of phagocyte to the affected area. Increased permeability helps phagocyte to migrate into the tissue and counteract with the pathogens. also a series of events. So, inflammatory mediator, once the site got injured, immediately it starts secreting the inflammatory mediators kind of histamines, kinins or prostaglandins, and three major events occurs in the inflammatory response. One is vasodilation, edema and influx of phagocytes. Vasodilation is nothing but the increased blood flow to the site, and thereby redness or swelling or increase in tissue temperature will happen. Edema is nothing but the accumulation of fluid which causes the swelling. And the third is the influx of, they are inviting the phagocytes to counteract with the pathogen. So, influx of phagocytes. So, they want phagocytes to come and act against the pathogen. Okay. So, influx of phagocytes, increased permeability helps phagocyte to the affected area and counteract with the pathogens with the series. So, this picture gives the series of events happening during influx of phagocytes. First, the adherence of phagocytes to the endothelial cell wall of the blood vessel, that is margination. And next is the extravasation of diapedesis, that the emigration between the endothelial cells to the tissue. First, phagocytes go and attach to the walls of the endothelial cells. Next, this is the extravasation, that is the between the endothelial walls it migrates, okay, between the endothelial cells to the tissue. The third one is migration through the tissue to the site of ear, site of infection. So, first influx of phagocytes, first it go and attach to the endothelial cell wall, and next extravasation occurs, like between the endothelial cell it will go and finally reaches the site. These are the three events, these are the three series of events happening at the influx of phagocytes, As the phagocytes accumulate at the site of action and begin to phagocytose the bacteria and release the lytic enzyme that may damage near the cells. The accumulation of dead cell or digested material and fluid forms a substance called pus. And this is the image of influx of phagocytes. And this is a very detailed image of an inflammatory response. So, these are the inflammatory mediators which I said, histamines, bradykinins, prostaglandins. These are the major cells involved, mediators involved in inflammatory response, a chemical histamine, kinins and bradykinins. In addition to the process of inflammation, it also helps enzymes involved in blood coagulation cascade to enter the tissue. Okay. So, this results in activation of clotting, which in turn helps in the wall of infection area and prevents spread of infection. Once the inflammatory response subsides, tissue regeneration process begins. Okay. So, there are two types of inflammation, acute inflammation and chronic inflammation. Acute inflammation happens only for a shorter period of time, whereas the chronic inflammation occurs for the prolonged period of time. So, lasts for a shorter duration, and main cells involved in acute are leukocytes, are predominantly neutrophils again. Frustrated phagocytosis, again it is the biomaterials is much bigger than the attached cell, release of products to degrade the biomaterial. So, this acute inflammation resolves within 1 week, whereas the chronic inflammation, it is less uniform than the acute inflammation. Presence of macrophages, monocytes and lymphocytes are the major cells involved in chronic inflammation. So, if you place a biomaterial inside our body, there are series of inflammatory responses, right. Acute inflammation, first is biomaterial. When an implant is placed inside the body, first like or injury or that is different, and when an implant is placed, acute inflammation, chronic inflammation, granulation, that is the formulation of granulation tissue, granulation, and the foreign body, that is the fibrous tissue, and the foreign body reaction. Okay, this foreign body reaction is mainly composed of macrophages and foreign body giant cells. So, this will forms a layer outside the implant. So, thick layer outside the implant will be formed, which is macrophages and foreign body giant cells. The detailed explanation of inflammatory response with respect to implant will be explained in later sessions. So, in this session we learnt about what is immunity and what are the cells involved in immunity and what are the organs of the immune system, as well as the few basic steps of inflammatory response. Thank you.